Unit 3.3, and we're actually going to start on page 58 today. But before we continue, I did want us to stop for a second and review. What have we talked about already? Well, so far, all we've talked about is Mendelian genetics. And in Mendelian genetics, we had traits or alleles that were dominant and alleles that were recessive. Okay? How did we represent those alleles? Remember that an allele is a different version of a trait. So if we kept it as something simple like our hitchhiker's thumb, remember, was a dominant trait, and so we could represent it with something like a big H. And then we had recessive alleles, a straight thumb, which is what I have, that we could represent with a little h. So these are the individual alleles. Alleles are different versions of one trait. But what about if we're actually describing somebody's genotype? What two alleles did they get from both of their parents? Well, if I was homozygous meaning that I have two of the same alleles, I could be homozygous dominant, or I could be homozygous recessive, little h, or big H for dominant. And if I was heterozygous, my zygote had two different alleles, then I would be big H, little h. If I'm looking at all three of these genotypes, well, which genotype would give me hitchhiker's thumb? It would be my homozygous dominant and my heterozygous, because the dominant masks the recessive allele. Okay? So let's just keep that in mind while we move into our new types of genetics. We're actually going to be talking about different types of inheritance. So Mendelian inheritance is the simple one thing is dominant over another. Hitchhiker's thumb is dominant over a straight thumb. But we have 5 more types. And these traits are not just dominant or recessive. Therefore, they do not follow the Mendelian genetics. So when we look at things that are non-Mendelian, this isn't going to be dominant versus recessive. There's going to be more stuff going on. So it does get a little more complicated. But if you use the notes and the way they're set up, we can keep them pretty easy, and it'll be easy to keep them separated from each other. Some traits that don't follow Mendelian genetics would be like sickle cell anemia, a disease that we'll talk about, where instead of having round red blood cells, you'll actually have red blood cells that are shaped like a sickle that you would use at a farm, or a crescent like the moon. Hair is probably not one of the best examples, but we are going to use hair, curly, straight, and wavy, as another example. It's not really a true example, but it's a good example to see in humans. And then another type that we'll be looking at is blood type. Blood type is another example of non-Mendelian inheritance. So, we have a lot going on here. The first type of non-Mendelian genetics is called co-dominance. In co-dominance, both of them are cooperating. Both cooperate. If you think about the word cooperate, operate meaning work. So both alleles are actually going to work together at the same time. Both alleles are equally dominant in a co-dominant inheritance and will be expressed in the heterozygous. So again, our example here is actually going to be red blood cells, normal red blood cells, and sickle-shaped red blood cells. Okay, let's go ahead and label that picture here. This is normal, and this is sickle. And so if they both cooperate, what we're really saying is that we have 2 alleles, one for normal blood, one for sickle blood. And if they're both cooperating, they can both show up together at the same time or in the same person. So the phenotype for codominance what is the heterozygote? Well, remember that's when you have two different alleles. Your genotype, you're actually going to use two different letters here. So you can use like an A and a B, but since we're talking about normal red blood cells and sickle red blood cells, we'll use an N and an S. But really you could use any two letters. Our example, sickle cell anemia, is a co-dominant disorder. This means that you'll have normal disc-shaped cells, an N, and sickle-shaped cells, a S. Three possibilities you do need to fill in this chart. This chart will help you work out your assignments. We have three genotypes. We have homozygous, NN, and we also have homozygous SS. Since they are both dominant, we really wouldn't need to say homozygous dominant and homozygous dominant, because they're both homozygous dominant. They're just homozygous dominant for two different things. NN, if you have both of those alleles from normal cells, that means you'll only have normal cells in your body. If SS, you have two alleles for sickle cells, then you'll only have sickle cells in your body. And NS, normal and sickle, literally means you'll have normal and sickle cells in your body. One benefit to this disorder, this disease, is that if you're heterozygous, NS, having both normal and sickle cells, you will actually be resistant to a disease called malaria. Malaria is a disease that is transmitted by mosquitoes. Could be very, very dangerous. You can be very sick from it. You can even possibly die from it. But one benefit to this disease, sickle cell anemia, is that it makes you resistant to malaria. Then we have our co-dominant example. What I actually want you to do is move down your notes a little bit in the summary box to work out this example. I do want you to first of all kind of split that summary box in half because we'll put two examples in the bottom summary box. You want to go ahead and label the left side co-dominant example, or just write co-dominant. And here's our example problem. In our example problem, it says, cross a person with normal cells with a person with only sickle cells. Well, if you're normal, you only have normal-shaped red blood cells. So you'd have to have an N. And what else do you need, look back at your chart, to be able to have all normal cells? Well, you would need a second N. And then you're going to have cross. Cross means like mate. You're crossing or fertilizing sperm with egg. A person with only sickle cells. So if you only have sickle cells, you're SS. Okay? So what I want you to write underneath codominant is I want you to write, let's say, person with normal cells plus person with sickle cells. So that we know when we look later on at our example that that's what we initially had. And so let's plug in those two genotypes. We'll do the normal person first, N, and we'll do the sickle cell person second, S. It really honestly doesn't matter if you write an S first or an N first. It doesn't matter because they're both dominant, so they're both going to show up. But usually, just to keep everything the same and everything organized, we just go with whatever's first in the alphabet, okay? So that way, when we're checking our papers, it'll make it a lot easier. So N comes first, so I'll put N, drop those down. This column, N goes down. And then next, S comes all the way over, and I'll write that second. NS, NS, NS. So what are the chances they have a child with no sickle cells? Well, after you're done doing your Punnett square, let's write child with no sickle cells. Let's give a percentage equals 0%. Okay? All of their children, no matter when boy or girl that they have will have normal cells and sickle cells. Remember that these boxes represent 25% chance. Okay, 25% chance for each box. But it's just really saying that if I have a child, 1 in 4 chances, or actually in this case, 100% chance that all of my kids would have normal and sickle cells. There's no other option there. Okay, let's go ahead and continue. Next we have incomplete dominance. Incomplete dominance is where neither allele is completely dominant to mask the other. You have a phenotype, the genotype. When you're talking about the phenotype, the heterozygous genotype results in a blended phenotype. For our genotype, well, how do we write this down? Capital letters for both alleles and use a prime symbol. Prime almost looks like an apostrophe. In humans, this happens in genes for curly hair. So we're going to use a regular capital H for curly hair, and we're going to use an H prime for straight hair. Notice there's that little prime there. So major thing here is that neither allele completely masks the other. So instead, they're going to blend or mix together, like it says in your phenotype. So for example, if I have HH, 2 alleles for curly hair, well, I'll have curly hair. If I have H prime, then I'm going to have straight hair. 2 alleles for straight hair gives me straight hair. But if I have a heterozygote, 2 different alleles in the same zygote, then I have 1 allele for curly hair, a regular H, and I have an H prime allele for straight hair. Well, curly hair plus straight hair blends together, and I think of that little prime as kind of a mixer in a blender, blends it together, and I get wavy hair. Okay? So make sure you take a minute to finish copying that down. But let's look at an example. It says cross a person with straight hair with a person with wavy hair. So if you have straight hair, what should you have? Well, straight hair was H prime. What was my second allele? Also H prime. Now if I have a person with wavy hair, then what was that? Well, that was curly hair, just a regular H, and H prime for the straight hair. Curly plus wavy equals straight. So then it says, what is the probability of them having a straight-haired child? Well, remember that in this case, since we have 2 different letters, or a letter with its prime symbol, we're actually going to write the regular H first. So I'm going to bring over my regular H, and then notice that all I have left are my H primes, so I can just go ahead and fill those in. So what is the probability of them having a straight-haired child? Well, what did you need to have straight hair? If this should be going in the right-hand side of your summary box, well, straight hair would be H prime, H prime. If each box represents 25%, well, where do I see H prime, H prime? Right down here. So 25% plus 25%. Well, for them to have a straight-haired kid, straight hair child, that's going to be 50% chance. And we're actually just going to stop there for today, and we'll talk about multiple alleles and sex-linked genetics later on.